All right, everyone. So let's wrap up chapter 16, shall we? This is going to be part two of that chapter, pages 565 to 591. And what we're going to do today is basically just go into depth and detail of how the signaling that we introduced in the last video actually happens. So last video was very conceptual, and this video is going to be much more practical, talk about how this happens at the cellular level. So we're going to start off with GPCRs or G protein coupled receptors and talk about how they transduce signals into the cell. That's going to bring us to second messengers. We're going to talk about two different stories of G protein coupled receptor second messengers, cyclic AMP as well as calcium. And then we'll go on to the other type of specific receptors that are responsible for signal transduction, enzyme-coupled receptors, and our example of that will be the RTKs, this huge receptor protein family, receptor tyrosine kinases. And then we'll wrap up cellular communication in general by talking about some big picture concepts. So let's talk about GPCRs. G protein coupled receptors are the largest family of cell surface receptors in eukaryotes, and that's hands down. Humans have more than 700 types of these proteins, and mice have over 1,000 dedicated G protein coupled receptors for olfaction alone. That's also true of other animals that have an amazing sense of smell, such as dogs. So you got 1,000 different receptor proteins just for the sense of smell, which is incredible. These kinds of receptors are sensitive to a very wide range of cellular signaling molecules, including hormones, paracrine molecules. Remember, the paracrine system is that local system where cells secrete a molecule nearby and other cells can pick it up. Neurotransmitters also often bind to G protein coupled receptors in the nervous system. These receptors play a role in every single aspect of cellular and organismal communication. Whenever the body needs to send a message through itself, whenever a cell needs to send or receive a message, largely those are going through G protein coupled receptors. Not exclusively, but G protein coupled receptors, again, are the most common and the largest family. The signaling molecules that bind to and interact with these receptors are just as varied as the receptors themselves. Again, these signaling molecules can be proteins, large proteins. They can be small peptides, which is just a very limited number of amino acids linked together. Signaling molecules that bind to GPCRs can be modified single amino acids, modified fatty acids. All of these are known to be ligands, or again, signaling molecules that bind to this receptor type. So hopefully it should be no surprise, even with this limited background, that these receptors are commonly the target of pharmaceutical drugs because they do so much in the body and because so much of how the body senses and reacts is through this receptor family. By targeting these receptors with pharmaceuticals, we can get a wide variety of effects in the body. In fact, 50% of all therapeutic drugs work through GPCRs. Half of all therapeutic drugs are designed to bind to this receptor type, showing um, just how ubiquitous this receptor is in our body. And maybe you've heard of them. We talk about them briefly in Bio 107, so maybe you've heard of them before and you have a little bit of memory, but maybe you haven't, and yet they're fundamental to all of cell biology. So let's talk first about what you could think of as their anatomy, their structure. G protein coupled receptors are transmembrane proteins, so they're integral. They have hydrophobic patches that interact favorably with the fatty acid tails of the cell membrane, and they have an extracellular domain. They have a portion that's at the cell surface, of course, facing outward, and that's the portion that can bind to the signaling molecule, and that's what we see here in light green. So extracellular domain that binds the signaling molecule. And then I've mentioned this before, even in our class, but we have 1, 2, 3, 4, 5, 6, 7 transmembrane domains. So G protein coupled receptors are a member of the larger 7TM protein family, which are membrane proteins that have 7 transmembrane domains. And then, of course, for the signal transduction to be possible, we have an intracellular domain. So, how did this protein get here? One might ask. And if one were to ask that, you would hopefully be able to remember that these proteins were, of course, first synthesized and born in the rough ER because they are membrane-bound proteins. And in the process of being made in the rough ER, they would be threaded through the ER membrane using start and stop transfer sequences. Then they were shipped to the Golgi for further processing through vesicles, and then shipped to the cell membrane via transport vesicles. This involved clathrin, RABs, tethering proteins, V snares, T snares. So I really want you thinking about this material holistically. I want you thinking about how these stories connect into a greater whole. Don't silo this knowledge. Let the semester tell you the story of life. So we know how proteins like this are born. We've talked about that in previous lectures, and you'll certainly be talking about that in your next e-report and your next mastery check. So you all know this. You've been exposed to this before. Again, G-protein coupled receptors, all of them belong to this larger superfamily of membrane proteins, the 7-pass transmembrane receptor protein superfamily, or 7TM for short. Members of this protein family include rhodopsin, which bacteria rhodopsin is a close cousin of, our own olfaction receptors for smell, a receptor in yeast that's responsible for sensing the yeast pheromone alpha factor, and so many other molecules that are responsible for cellular communication. Evolutionarily speaking, G protein coupled receptors are ancient, and again, even bacteria have close cousins of these proteins, such as bacteria rhodopsin, which we've already talked about. So let's talk about how these receptors work. And the way I'm going to try to handle this is I'm going to give you a relatively quick superficial overview and then retell the story in much more detail. So be prepared to hear this twice, and that's simply to give you the context in the shortened version and then fill in the details later on. I'm afraid if I just went into the detailed explanation, it would be too many moving parts and you wouldn't have the context for this material to fit into. So as we already know from Monday's lecture, the first thing that happens to any receptor when a signaling molecule binds to it is there's a conformational change. The signaling molecule binds to the receptor changes its shape to accommodate that binding, and that causes a conformational change in the receptor. In the case of a G protein coupled receptor, this conformational change in the receptor, that again occurs because the signaling molecule was bound, activates the associated G protein. This is why these are called G protein coupled receptors. They're receptors that are coupled to a separate G protein. And the G protein is activated when the signaling molecule binds to the receptor and the receptor changes shape. So what do I mean by activates a G protein? Well, there's a wide variety of different G proteins out there, which interact with a very wide variety of receptors. And all these G proteins do a wide variety of things. But all G proteins have one thing in common, and it's why they're called G proteins. They bind to guanosine triphosphate and guanosine diphosphate. They bind to these guanosine phosphates. Every G protein is made up of 3 subunits, an alpha subunit, a beta subunit, and a gamma subunit. And it's the alpha subunit that actually binds to GTP or GDP. So now here we're starting to see more of the story. Here's the G protein coupled receptor. But when I say that, the G protein coupled receptor, I just mean the receptor. So the receptor is the 7 transmembrane protein. And here it is. Here's the receptor. But in any functional context, this receptor is going to be coupled to a G protein. And here's the G protein. The G protein that's made up of an alpha, beta, and GABA subunit. And because it's a G protein, it can bind to GDP or GTP. In this case that you're looking at here, the G protein is bound to GDP, and that's why it's in its inactive form. So G proteins are inactive when they bind to GDP. So in this case, in this picture, the G protein is dormant. It's not doing anything. Remember what I said a moment ago. When the signaling molecule binds to the receptor undergoes a shape change to accommodate that binding, and the shape change then activates the G protein. Well, what do we mean by activate the G protein? We mean let go of that GDP and pick up a GTP. So first thing that happens to any receptor is a signaling molecule binds to it. That causes the receptor to undergo a conformational change. This conformational change in the receptor activates the G protein. That's the G protein that's associated with that G protein coupled receptor. Again, you can think of this as the signaling molecule binds to the receptor and kind of forces and elbows its way into it. And that forcing and elbowing of the signaling molecule nudges side chains of the receptor. So the side chains of the receptor kind of move, they have their conformational change, And that conformational change of the receptor becomes large crowds of nudging elbows into the ribs of the G protein. And when the receptor gives the G protein a swift elbow to the ribs because it just underwent its own conformational change, the G protein is going to let go of GDP, pick up GTP, and become active. And now the G protein is active. So we can kind of deduce this a little bit. Why is the G protein active right now? Because it's bound to GTP, guanosine triphosphate. Well, why did the G protein bind to guanosine triphosphate? Because it was elbowed in the ribs by the receptor. Well, why did the receptor undergo a conformational change? Because the signaling molecule bound to it. So all of this is happening because of the presence and binding of the signaling molecule. G proteins that are bound to GTP are active, but that's only happening because of the signaling molecule binding. Once the G protein is active, the alpha subunit separates from gamma and beta, and the alpha subunit can alter other target molecules there at the cell membrane and change the way that the cell behaves, change what happens in the cell. And this initiates one of those signaling cascades that we talked about in the last video, a cascade or flow of changes that propagates into the cell, and there is your signal transduction. So far that covers how this system is flipped on. Signaling molecule binds, receptor changes its shape, G protein gets a nudge to the ribs, and it picks up GTP. But signals must also be turned off, right? I've used this analogy before in this class, but if you get a phone call, the phone call is that the cable repairman is coming tomorrow, and you take that message and you give it to your friend. You say, hey, Rumi, cable guy's coming tomorrow. Hey, Rumi, the cable guy's coming tomorrow. Hey, Rumi, the cable guy's coming tomorrow. And if that message, if that signal transduction never gets turned off, sooner or later it's going to become tomorrow, and you're going to be saying the cable guy's coming tomorrow. Not true anymore. The cable guy's coming today. So for messages to be appropriate, they have to be received, transduced, and then turned off. Otherwise, they become inaccurate and inappropriate. So to accomplish this at the cellular level, G proteins have themselves enzymatic activity. More specifically, they have GTPase activity. Well, what does GTPase activity mean? It means that G proteins can hydrolyze GTP to create GDP. And as we said before, once G proteins do that, they hydrolyze the GTP they're holding and they create GDP. Well, that leads to the inactive form of the G protein. So G proteins turn themselves off. When the G protein hydrolyzes the GTP it picked up, leaving GDP, guanosine diphosphate, it is now bound to that GDP, and therefore that G protein is now inactive. And so what happens is the G protein is going to reassociate with the receptor. And if the signaling molecule is still bound to that receptor, the receptor is going to give that G protein another swift elbow to the ribs. The G protein is going to let go of GDP, pick up GTP, and go back to doing its signal transduction job. And that's appropriate because the signaling molecule was still bound to the receptor. If instead the inactive G protein goes back to the receptor and the receptor is dormant because the signaling molecule is gone, well, then we want that message to stop. That message is no longer valid, and the G protein will remain dormant and inactive at the receptor, waiting for the cycle to continue again. So essentially, the G protein is on a timer. It's only active for a brief time, and then it converts GDP into GTP, and it turns itself off. And then it goes back to the receptor and checks for the validity of the information. If the signaling molecule is still there, the G protein will be turned on again. If the signaling molecule is gone, the system will remain off. So what screws up this system? Well, to drive home the point that this timer mechanism is critically important, that being turned off is just as important as being turned on for signal transduction, let's take a quick little detour to see what happens when this turning off system is blocked, when the GTPase system is blocked. The bacteria that causes cholera secretes a molecule that's called cholera toxin, not surprisingly. And this molecule enters the cells of the intestine. And when cholera toxin is absorbed by intestinal cells, It specifically modifies the alpha subunit of a G protein so that that alpha subunit can no longer hydrolyze GTP. In other words, that subunit remains stuck on in the presence of this toxin. In this case, the stuck on G protein keeps a chloride channel open. So chloride keeps rushing into the cell. And remember, when ions rush into a cell, what follows? We talked about this just a couple weeks ago. What follows ions, what follows solute is water. So water comes rushing in. So we keep losing chloride, so it flows out of the intestinal cells, and the water follows. Since the water is following the chloride out of the cells, the water is flooding the intestine. And what happens when water floods the intestinal system? Well, we get diarrhea and dehydration of the cells. So the reason why acute cholera causes diarrhea and dehydration is because this G protein won't turn itself off. If not treated, this can become life-threatening due to dehydration, and it's all due to a single G protein not being able to hydrolyze its GTP. Whooping cough is caused by a very similar toxin in a very similar mechanism, but in the lungs, which is what creates so much mucus and fluid in the lungs of somebody with whooping cough, again, due to nothing more than a single G protein stuck in the on configuration. Okay, so off the detour and now back to the main story here. So great, now we understand how G proteins are turned on and they turn themselves off, but what do G proteins actually do when they're on? Well, what's implied by the cholera story is that G proteins can open ion channels. And sure, that's true. That's one of the things they can do. In essence, what G proteins do when they're activated is they turn on, or in the case of a channel, you can think of that as open, but they turn on membrane-bound proteins. That's all they do. So when a G protein becomes active, it activates some other protein. If that protein happens to be an ion channel, then that ion channel will be open. But G protein coupled receptor controlled enzymes do something even more profound in the cell. Here are some specific examples. Your heart rate is controlled by two nerves. One nerve, when it fires, speeds the heart rate up, and the other nerve, when it fires, slows the heart rate down. And through the competing opposing actions of these two neurons, you can achieve homeostasis and have a regulated heart rate. When the nerves that slow the heart down fire, they release the neurotransmitter acetylcholine. And we actually talked about acetylcholine before, specifically in regards to the cardiac muscle. When acetylcholine is dripped on cardiac muscle, it relaxes the heart, and this is what slows the heartbeat down. Acetylcholine binds directly to a G protein coupled receptor on the surface of the cardiac muscle cells. And when that binding occurs, the activated G protein coupled receptor opens a potassium channel. So potassium goes rushing through the channel. As potassium leaves the cardiac cell becomes hyperpolarized. It gets more and more negative inside the cell. And since the cell is hyperpolarized, that's going to make it so that a lot more sodium is necessary to enter the cell for an action potential to be fired. In other words, it makes it much harder for the neuron to fire and cause the heart muscle to contract, so thus slowing it. So another example of how just a G protein coupled receptor, and the G protein it's coupled to, having a dramatic effect on the cell. But again, just another ion channel story. To me, what's much more interesting than the ion channels are the enzymes that G proteins can affect at the cell membrane. And this is where we get those very loud, amplified, dramatic responses from the cell. These systems tend to be more complex and more cascading. The two most common enzymes that can be activated by an activated G protein are adenylate cyclase and phospholipase C, and they are completely different signal transduction pathways, and we'll talk about them separately. These two enzymes are activated by different types of G protein coupled receptors and therefore activated by different types of G proteins, but they are the two examples that we'll discuss. Adenylate cyclase as an enzyme catalyzes a very straightforward and simple reaction. It converts adenosine triphosphate into cyclic adenosine monophosphate. And this is what that reaction looks like. So you remove two of the phosphates and you go down to the monophosphate, but then you make this funky bond between the phosphate and the 3' hydroxyl. And that's why it's cyclic AMP, because we've made this cyclic bond right here. Cyclic AMP or CAMP is always a signaling molecule. It carries information. Phospholipase C catalyzes a more complicated reaction, which we'll talk about when we get to that enzyme. In either case, the G protein is indirectly responsible for the production of intracellular signals, and those signals then change the cell's behavior, and the change in cell behavior is in direct response to the message it received at the receptor. So what's happening? What's happening is a signaling molecule bound to a G protein coupled receptor. As a result, the G protein coupled receptor had a conformational change. As a result of that conformational change, it nudged the G protein, and the G protein let go of GDP and picked up GTP. That activates the G protein. Then one of the subunits of the G protein, in this case the alpha subunit, migrates over to another membrane-bound enzyme and activates it. Now this enzyme is on. And a result of this enzyme being on, it's going to take precursors and catalyze the conversion of them into second messengers. This could be ATP, and the enzyme is making cyclic AMP as a result. These small second messenger molecules are then going to diffuse into the cell and indirectly cause a dramatic behavioral change of the cell. And that behavioral change of the cell is the change we want in order to respond to the signal we just received. Again, these secondary molecules are second messengers, and we typically refer to the signaling molecule as a primary messenger, as the first molecule to deliver the message. All of that stuff, the G protein coupled receptor, the G protein itself, the enzyme, in this case adenylate cyclase, all of that stuff is membrane bound. It's stuck at the interior face of the cell membrane. But for signal transduction to be successful, we're trying to get information into the cell. And that's why we need the second messengers, because the second messengers are small and diffusible. So the second messenger carries the message into the cell. And in this example, the second messenger is going to be cyclic AMP. Cyclic AMP is not like DNA or RNA or even protein. There's no information in the molecule. It is merely the presence of the molecule that carries information. So a good metaphor analogy for this that I can think of are old Native American smoke signals. It's not as though when Native Americans were sending smoke signals from one group to another, the smoke itself had any information in it. It's not like it was a pattern of smoke or somehow the Native Americans figured out how to encode information in the smoke. No, it was if you see smoke, there's danger. Prepare for battle. And if you don't see smoke, there's no danger. Don't worry about battle. It was just the presence of smoke that carried information, a very binary system. And such is the case with cyclic AMP. Usually cyclic AMP, its presence activates a single other protein. And in the scenario we'll discuss, that's protein kinase A. That's protein kinase A. So my genuine hope is that you already have some idea of what this enzyme is going to do when it's active. It's going to phosphorylate something. It's a kinase. So when cyclic AMP is present and it activates protein kinase A is then going to phosphorylate other target proteins. And remember, phosphorylation for proteins is a toggle switch. Proteins that are on can be switched to off through phosphorylations, and proteins that are off can switch to on, which is due to phosphorylation. So it's protein kinase A that achieves the actual physiological response. The cell's behavior changes because a whole slew of proteins that were on are now off, and a whole slew of proteins that were off are now on. That's what changes the cell's behavior. But what toggled the switches on all of those proteins? Protein kinase A. Why was protein kinase A active in the first place? Because cyclic AMP bound to it. Why was cyclic AMP around? Because the adenylate cyclase enzyme made it. Why is adenylate cyclase on? Because it was activated by a G protein. Why is the G protein active? Because it's bound to GTP. Why is the G protein bound to GTP? Because the G protein coupled receptor nudged it. And why did the G protein coupled receptor nudge the G protein? Because it was bound by the signaling molecule. So the signaling molecule binds to the receptor. The receptor causes the conformational change in the G protein. The G protein picks up GTP and activates the enzyme adenylate cyclase. Adenylate cyclase is going to convert ATP into cyclic AMP. Cyclic AMP is going to diffuse into the cell as a second messenger and activate protein kinase A. Active protein kinase A is going to phosphorylate target proteins, and those target proteins that were phosphorylated are going to ultimately change the cell's behavior. And if the signal was adrenaline, we need energy in a hurry because we're looking at a tiger, then the behavioral change in the cell is breaking down glycogen to release tons of glucose to liberate that energy and respond to that tiger. Ligand binds, G protein is nudged, G protein picks up GTP, G protein migrates to adenylate cyclase and turns it on. Adenylate cyclase takes ATP and converts it into cyclic AMP. Cyclic AMP diffuses into the cell and turns on protein kinase A. Protein kinase A, now active, phosphorylates target proteins. And now that those phosphates are added and those proteins are toggled, we have a change in the cell's behavior. Ah, magnifique. Sometimes protein kinase A, once active, can actually be transported into the nucleus. How might that happen, you ask? Well, I'm glad you asked that question. They're transported into the nucleus because a nuclear transport receptor recognizes that active protein kinase A and escorts that protein kinase A through the nuclear pore complex. We talked about that too. And then once in the nucleus, protein kinase A can also have effects typically on gene expression, turning genes that were off on or turning genes that were on off. So table 16.3 from the textbook gives you some common signaling molecules that bind to G protein coupled receptors, the target tissue where that binding occurs, and the responses, the physiological responses that occur. But all of it is through this kind of system. So with that air quote easy story of signal transduction through adenylate cyclase, let's talk about the more complicated signal transduction story relevant to G protein coupled receptors, the one that uses calcium as its second messenger. So calcium serves as a second messenger in the other commonly used G protein coupled receptor system that we discuss in this class. And this system uses a whole bunch of different molecules, but one of the primary molecules is a membrane component called PIP2. The full name of PIP2 is, let's see how I do, phosphatidylinositol 4,5-bisphosphate, but for you and me, this is a phospholipid. It's a membrane component. It's a specific phospholipid, but it's a phospholipid. So of course, this story starts with a primary messenger binding to the receptor on the outside of the cell. The receptor undergoes a conformational change, which nudges the G protein. As a result of that nudge, the G protein lets go of GDP and picks up GTP and becomes active. But now, instead of going to adenylate cyclase, the active G protein is going to migrate to a membrane-bound enzyme called phospholipase C. What do you think phospholipases do? Well, phospholipases cleave phospholipids. So that PIP2 phospholipid is now going to be cleaved by the active phospholipase C. Once we cleave PIP2, we yield its two halves, its two molecules. So PIP2 is cut in half into two molecules, one called DAG, our old friend DAG, and the other called IP3. IP3 is a second messenger. It diffuses into the cell. It actually travels to the ER membrane, and it's going to open calcium channels. Calcium is going to come rushing out of the endoplasmic reticulum through those open channels. DAG is going to remain in the membrane and activate protein kinase C, a membrane-associated protein kinase. Protein kinase C, just like protein kinase A, is now an active kinase. It's going to phosphorylate lots of things. But one of the things that protein kinase C is going to phosphorylate are calcium channels. So calcium is going to come rushing into the cytoplasm from outside due to the protein kinase C opened channels. And calcium is also going to come rushing into the cytoplasm through the endoplasmic reticular stores. Because like I said before, IP3 opened those channels. So you got calcium rushing into the cytoplasm in two ways. Through cell membrane channels that were opened by protein kinase C and through ER channels that were opened by IP3. But before we go forward, I want to do that kind of reverse explanation again. Why are the membrane channels open? Well, they're open because protein kinase C phosphorylated them. Well, why is protein kinase C active? Because it was bound by DAG. Why is DAG around? Because it's one of the cleavage products of PIP2. Why was PIP2 cleaved? Because phospholipase C cleaved it. Why was phospholipase C active? Because the G protein turned it on. Why was the G protein on? because it was bound to GDP. Why was it bound to GDP? Because it was nudged by the G protein coupled receptor. And why did the receptor do that nudging? Because it was bound by the signaling molecule. And let's not forget that also calcium channels at the ER membrane are open. Why are they open? Because the other cleavage product of PIP2, IP3, bound there and opened those channels. So now you got calcium rushing into the cytoplasm. Calcium is the second messenger. It's small and it's diffusible. What does calcium do? Well, calcium binds to a protein called calmodulin. In fact, that name, calmodulin, means modulated by calcium. When calcium binds to calmodulin, active calmodulin can then bind to the calcium modulated kinase or CAM kinase, and that kinase is turned on. Sound familiar? And once that CAM kinase is on, it's going to phosphorylate a whole slew of proteins in the cell, turning some of them off and some of them on. You're going to get a massive change in cellular behavior that is in direct response and physiological relevance to the message received by the primary signaling molecule. Now, these signaling cascades are ridiculously fast. The nervous system is always going to be the fastest mode of communication in the body because it's built on electrical impulses. Nothing moves faster than ions. But the G protein coupled receptors, the speed of them is no joke at all. Think of how quickly your heart rate accelerates after being scared, right? Like the most profound example for this for me is consistently, not that I'm a bad driver, not that I'm a risky driver, not that I drive too fast, not that I drive too fast. But when I have a close call on the road, it's like I've barely processed what happened and my heart is racing, right? I haven't even had time to cognitively appreciate the danger that I was in, yet my heart is already racing. That's the action of G protein coupled receptors. The sense of smell is all G protein coupled receptors. And think of how quickly you smell and process something. The salivation response is G protein coupled receptors. Even sensing light in the eye is mediated primarily and initially by G protein coupled receptors. The fastest G protein coupled receptor responses, and that's the response to light, are 20 millisecond lap time. 20 milliseconds, that's 2/100ths of a second. It takes 2/100ths of a second for light to hit your eye and then be conduced or transduced by a G protein coupled receptor. Like I said before, yeast sense and respond to their environments, specifically pheromones in their environment through G protein coupled receptors. And we sense and respond to our environment in the same exact way through G protein coupled receptors. This is an evolutionary legacy. Evolutionarily, the way to sense the world has always been mediated by G protein coupled receptors. Once life became multicellular, sensing meant something very different than it means to a yeast cell. Sensing became light detection. Sensing became olfaction. Sensing became much more complicated, but it still used the same toolkit. It used the toolkit of G protein coupled receptors, which I find truly amazing. So that leads us to enzyme-linked receptors, our second class of specific receptors that we're going to talk about in this class. Enzyme-coupled receptors differ from the 7-transmembrane G protein coupled receptors in many, many different ways. But the primary way is that the enzyme, the kinase, that is going to be used to transduce the signal into the cell is coupled to the receptor for this receptor class, and that's why they have their name. So if you think about the GPCRs, it was the second messenger that diffused into the cell and turned the kinase on, and that kinase gave us our cellular response. For enzyme-coupled receptors, the kinase is linked directly to the cell, to the receptor itself. When activated, the kinase that's coupled to the receptor is going to set a cascade into motion, and that cascade of cellular events is going to transduce the message into the cell so that the cell can respond accordingly. Enzyme-coupled receptors are usually a dimer of two identical subunits. And typically what happens is that the signaling molecule serves as the bridge to bring those two subunits together. Each subunit has like a half binding site for the ligand. And when the ligand is present, Each half binding site is brought in close proximity, and now the receptor is made whole. So it's pretty unique for enzyme-coupled receptors. The receptor exists in two parts, and it is the ligand itself that bridges those two parts and makes them one. As these two subunits move together and are bridged together by the signaling molecule, that's going to kick off the signaling pathway. For tyrosine receptor kinases, there are tyrosine residues, tyrosine amino acids, in the intracellular domain of the receptor. Again, this is where these receptors get their name, receptor tyrosine kinases. Kinases that contain tyrosine that are linked to receptors. Then, sorry, having some technical difficulties here. All right, I think we're good. I got some weird message about my microphone, but hopefully this video will keep recording audio. Then we can activate the receptor. So the receptor begins in an inactive form because the signaling molecule was not present. But once these two receptor halves are bridged together, then we get one half of the receptor activating the other, activating it through phosphorylation. So we actually call this autophosphorylation. Auto meaning self, and phosphorylation meaning that, phosphorylation. So these receptor tyrosine kinases are self-activating in the presence of their primary messenger. Primary messenger brings the two halves of the receptor together, and then one half phosphorylates the other, and that half phosphorylates the first. The receptor autophosphorylates, and it becomes active. Binding of the signaling molecule on the extracellular domain is what resulted in this bridging and this autophosphorylation. So this is a direct result of the primary messenger. And then once those phosphate groups are added to those tyrosines on the intracellular domain, those phosphorylated tyrosines serve as docking sites for, and here I'll use the weird language of the textbook, for a zoo of proteins. That's what the textbook says. What do we mean by a zoo of proteins? We mean a lot of different proteins. So a lot of different proteins bind to phosphorylated tyrosines, and they're only binding to the receptor now because the receptor has autophosphorylated. Well, why did the receptor autophosphorylate? Because the two halves of the receptor were brought together, and what brought those two halves together? The presence of the signaling molecule. Once all of these recruited proteins are bound to the receptor through this bridge of the phosphorylated tyrosines, we can transmit the signal into the cell, and we can coordinate a lot of different cellular responses. So phosphorylated proteins are extremely stable. How do we turn receptor tyrosine kinases off? Again, how do we avoid falling into the cable guy is coming tomorrow trap? How do we make sure that we deactivate this system so that we don't give an inappropriate message that's no longer valid? Well, there are protein tyrosine phosphatases. Remember what phosphatases do? They remove phosphate groups, and these protein tyrosine phosphatases specifically recognize phosphorylated RTKs and remove the phosphate groups from them. In some cases, activated receptor tyrosine kinases, as well as activated G protein coupled receptors, are inactivated through destruction. They're targeted for destruction by getting certain chemical groups added to them. They're chemical groups called ubiquitin, if you're interested in the details. And once a protein is ubiquitinated, it's going to be brought back into the cell through pinocytosis. The vesicles that contain those membrane proteins are going to be sent to the lysosomes. And of course, the lysosomes will digest them down into amino acids for recycling. Different activated RTKs are going to recruit different proteins to their phosphorylated intracellular region. And so different receptor tyrosine kinases can cause different cellular responses. However, many of these receptors do seem to activate our old friend, well, maybe our new friend, phospholipase C. And phospholipase C, once active, is going to cut PIP2. PIP2 is going to become IP3 and DAG. DAG is going to turn on protein kinase A and open calcium channels. IP3 is going to diffuse into the cell and open calcium channels at the ER. And once calcium comes rushing in, it's going to bind to Calmodulin. Calmodulin is going to bind to CAM kinase, and CAM kinase will be turned on, and we'll get a massive kinase-driven response in the cell. At the end of the day, despite all the complexity, cells really have a relatively few set of tricks up their sleeve. And one of these tricks is the phospholipase C trick. to get things done in the cell. The other most common culmination point for receptor tyrosine kinases is the activation of a protein called RAS. And I want to be very, very clear here. I'm about to go into a lot of detail on RAS, and I'm really doing this for two reasons. One, as a case study for how involved these signal transduction pathways actually become. Two, because for any of you who go into medicine or to graduate school, you're going to be hearing about RAS and MEEKs a lot because they are a primary driver of cellular development, um, cellular differentiation, and cancer. So it's good to know a little bit about RAS from undergrad, um, even if you don't get all of the details. But all of that said, I'm going to hold you to a very superficial understanding of RAS, and I wouldn't worry too much about the details. Just try to get conceptually what's happening here. So EGF signaling, EGF is epidermal growth factor. So EGF signaling serves as an example of receptor tyrosine kinase signaling that specifically talks to RAS and uses RAS to transduce its signal into the cell. EGF is a small peptide, so it's a small chain of amino acids, and it does what its name suggests. It stimulates the growth of epidermal and epithelial cells, cells that line our body. The EGF receptor is, in this case study, of course, no surprise, a protein tyrosine kinase, and it self-phosphorylates. So it's an enzyme-linked receptor. It's a receptor tyrosine kinase. The EGF receptor subunits exist as identical monomers separated in half. When EGF binds, it's going to bridge those two monomers together. Those two monomers can autophosphorylate and signal can be transduced. So it's the dimer of the receptor. It's the dimer of the signaling molecule that brings the receptor together. Although EGF receptors self-phosphorylates or autophosphorylates, it's not this autophosphorylation that's required to activate the kinase. The phosphorylated tyrosines serve as docking sites, just as we said before. And these docking sites are going to recruit specific proteins. One docked protein recruits another, and this one recruits RAS and activates it. So here's EGF, the dimer of EGF. The dimer of EGF is going to bridge the dimer of the receptor. Once the dimer of the receptor is together, it will autophosphorylate, but it's not the active kinase that's going to bring the signal into the cell. All we needed was these tyrosines being autophosphorylated. One of these phosphorylated tyrosines is going to recruit and here adapter protein, and then that adapter protein is going to recruit RAS activating protein, the protein that activates RAS. What is RAS? Hold on. RAS is a G protein. G proteins bind to GDP and GTP. G proteins are inactive when they're bound to GDP, and they're active when they're bound to GTP, and such is the case for RAS. So these small G proteins like RAS work very much like the other G proteins that we've talked about. It's just that they don't have the the gamma and the beta subunits. They work alone. When RAS activating protein is recruited to the receptor, it is going to function as a guanine nucleotide exchange factor. It's going to act as a GEF. Well, let's break that down. Guanine nucleotide exchange factor. This sounds like a protein or a factor that causes something else to exchange its guanine. Okay, well, that's exactly what we want RAS to do. If RAS is a G protein and it's off, meaning it's bound to GDP, we want RAS to exchange its guanine nucleotide for GTP and be activated. And that's what activates RAS. So RAS is activated by this GEF, this guanine nucleotide exchange factor. When RAS is activated, it's going to change its conformation, it's going to bind to other things, it's going to activate other things, and one of the things that RAS is going to activate is RAF. RAF changes its conformation, and RAF is an active kinase. However, RAS and RAF are anchored to the membrane, and we need the signal to be transduced into the cell. So the active kinase RAF is going to do what kinases do. It's going to phosphorylate MEEKs. MEEKs are mitotic factors. MEEKs are going to activate extracellular signaling. MEEKs are going to activate extracellular signal regulated kinases, or ERKs. That's just a fancy name for a kinase that's turned on by some extracellular signal. Well, the extracellular signal here is the growth factor, EGF. And then the ERKs are going to activate transcription factors and other kinases. Genes will be turned on, proteins will be turned on, and we get a cellular response. So here's the flow chart for this specific example. EGF is the primary messenger. It's the signaling molecule. It's going to bind as a dimer to the EGF receptor. The EGF receptor is a receptor tyrosine kinase. The EGF receptor is going to be made whole and bridged by EGF binding. That receptor, once made whole, is going to autophosphorylate, and tyrosines on the receptor's intracellular domain will be phosphorylated. Those phosphorylated tyrosines are going to serve as docking sites for adapter proteins. And then some of those adapter proteins are going to recruit a guanosine exchange factor, a GEF. That GEF is going to force RAS to let go of GDP and pick up GTP. RAS is now activated. Once RAS is activated, it's going to interact with and turn on RAF, a kinase. Once RAF is activated, it's going to phosphorylate MEEK. MEEK is going to phosphorylate an ERK, and then the ERK is going to see to the cellular response. The ERK is going to phosphorylate transcription factors and other kinases, and the cellular response will change as a result of EGF binding. So as you can see, most of the activation in this pathway is due to kinases, it's due to phosphorylation. So we can expect that phosphatases are involved in turning all of this off because those phosphates need to be removed. Interestingly, the phosphatases we need to turn everything off are also initiated at the beginning of the cascade. And so termination and initiation both start together. It's just that termination takes a little longer. Those enzymes are a little bit less efficient. So cells are turning this system off just as they're turning it on, making the signal transduction very short-lived. And RAS, like other G proteins, of course, has its own GTPase activity, and it will turn itself off on a timer. Once it cleaves GTP to make GDP, it will inactivate. And there are other proteins called GAPs, GTPase activating proteins, which can accelerate that timer and make RAS turn off more readily. Turning off RAS is central to turning off cell growth. And so, not surprisingly, mutations in RAS are oncogenes, which is why I wanted to tell this story. Oncogenes are genes, mutant genes, that cause cancer. They're dominant alleles. You only need one mutant oncogene in order for cancer to be high risk, unlike tumor suppressor genes, which are recessive. So a lot of cancers trace back to RAS mutations. RAS and another protein called P53 are responsible for the vast majority of all human cancers, even though it's just two proteins. And it's because they play these central roles in cell growth, mitosis, and proliferation. So again, as a reminder, I know the details on that RAS story are heavy. If you want me to review it in class, I'm happy to, but I'm sharing it as a case study of one, how complicated these signal transduction pathways can get, and two, how fundamental and critical they are to understand both as cell biologists, the shoes we're in, and as medical practitioners or research practitioners in the future, because the mutations in these proteins can have such detrimental effects. So let's tie this all together, shall we? We've got three different stories really that we told here. One of them, I should say two of them, are the G protein coupled receptor stories. And then we've also got the enzyme linked receptors, the tyrosine kinase receptor story as well. For G protein coupled receptors, the primary messenger binds. As a result of that, we have a conformational change in the receptor. How sick of you are you of hearing me say this? The conformational change in the receptor is going to cause the G protein to let go of GDP and pick up GTP, and now the G protein is active. What can active G proteins do? Well, for some GPCRs, the activated G protein is going to migrate to adenylate cyclase. Adenylate cyclase is going to turn ATP into cyclic AMP. Cyclic AMP is going to diffuse deeper into the cell and bind to and activate protein kinase A. And protein kinase A is going to bind to and phosphorylate target proteins, changing the behavior of the cell. See, as complicated as it seemed before, it's not nearly as complicated now. For other G proteins associated with different G protein coupled receptors, once the G protein is activated, it's going to migrate over to phospholipase C. Phospholipase C is going to cleave PIP2, a phospholipid, into two halves, DAG and IP3. DAG is going to bind to and activate protein kinase C, Protein kinase C is going to phosphorylate and open calcium channels, and calcium is going to come rushing in from outside the cell. Meanwhile, IP3 diffused into the cytoplasm, and it bound to calcium channels in the smooth ER. And so calcium is going to come rushing out of calcium stores from the smooth ER. Regardless of where that calcium flowed in from, whether it flowed from outside the cell or from the ER, that calcium is going to bind to calmodulin. Activated calmodulin is going to bind to calcium modulated kinase or CAM kinase. And once CAM kinase is on, it's going to phosphorylate a host slew of proteins, changing their behavior and thus eliciting a cellular response. However, we said that sometimes when enzyme-coupled receptors, specifically receptor tyrosine kinases, are activated, they can also lead to the activation of phospholipase C, and that same cascade of calcium as a second messenger can happen as a result of an activated receptor tyrosine kinase. And that's an example of crosstalk, where multiple pathways can converge on specific enzymes or signaling molecules and fine-tune each other. But we also told this story of where sometimes for receptor tyrosine kinases, one of these phosphorylated receptors can serve as a docking site for a GEF, a guanine exchange factor. That GEF is going to force RAS to let go of its GDP and pick up GTP because RAS is a G protein. Active RAS is going to turn on a MEEK. MEEK is going to phosphorylate an ERK, and an ERK is then going to behave as a widespread kinase, phosphorylate a lot of target proteins, and change the cell's behavior. A lot of commonalities in these pathways, and a lot of differences, and a lot of complexity. And hopefully it'll be no surprise to you that, of course, these signaling pathways are going to be one of the topics that are in the next e-report. So, to summarize what we talked about in this lecture, I think it's a short one in terms of time, but man, oh man, is it a dense one in terms of content. We began by talking about G-protein coupled receptors as members of the 7-transmembrane superfamily, and they're found embedded at the cell surface. They're G-protein coupled receptors. So the receptor exists as a protein, but that protein is coupled to a G protein, and all G proteins have one thing in common. They are called G proteins because they bind to guanosine, guanosine triphosphate or guanosine diphosphate. When bound to GDP, the G protein is inactive, and when bound to GTP, the G protein is active. Once a G protein is activated and it picked up GTP as a direct result of being nudged by the ligand-bound receptor, the alpha subunit can target some molecules. The beta and GABA subunits can do the same independently. And the G protein eventually will turn itself off due to its GTPase activity. The two most common enzymes, at least the ones that we spoke about here in this lecture, that are activated by G proteins are adenylate cyclase and phospholipase C. Quickly, adenylate cyclase converts ATP into cyclic AMP. Cyclic AMP is the small diffusible second messenger. It binds to protein kinase A, and once active, protein kinase A is going to change the cell's behavior by phosphorylating a whole bunch of target proteins. If a G protein turns on phospholipase C is going to hydrolyze the phospholipid PIP2. PIP2 breaks down into IP3 and DAG. DAG is going to turn on protein kinase C, which allows calcium into the cell. IP3 is going to allow calcium into the cytoplasm from the ER. Once calcium is in the cell, it's going to bind to calmodulin. Calmodulin is going to activate CAM kinase, and we're back to this. You turn on a kinase, you're changing the cell's behavior story. Enzyme-coupled receptors, including receptor tyrosine kinases, have the kinase enzyme coupled to the receptor. The kinase is part of the receptor structure. Oftentimes, these enzyme-coupled receptors, and specifically for receptor tyrosine kinases, they're self-activating. The two halves of the receptor are bridged together in the presence of their signaling molecule, and once brought together and made whole, the receptor is going to autophosphorylate. Once the tyrosines of that receptor have been phosphorylated by the other subunit's kinase, it's going to be a recruitment site for many, many other proteins, oftentimes other kinases, that will then be activated and launch a signaling cascade of kinase of phosphorylation activity into the cell. RTKs are commonly activate phospholipase C as well, and then we crosstalk into our calcium as a second messenger story, and we did use the RAS small g protein story as a case study of other ways that these receptor tyrosine kinases can work. Whew. So that is signal transduction. We don't have much left, folks. We only got 3 weeks left of instruction in this class after we get through this material in the classroom. Next week, we'll be talking about the cytoskeleton, broken up over 2 lectures. The week after that, we'll begin talking about the cell cycle, and then on Wednesday, we'll talk about mitosis, and then we have cellular communities and cancer. But that's it. 6 more videos, 3 more weeks of instruction, and then this semester will be in the books. But let's not get ahead of ourselves. Before we start making plans for the summer, please let me know what from this video you would like me to review in the classroom. Thanks so much for watching.